So, in a way, this is actually your first baby picture, the moment the sperm penetrates the egg. And if you think a little more deeply about this random sperm could have been some other random sperm, and you might be completely different. The process whereby which the sperm and the egg are made is known as meiosis. That's what we'll look at in this video. Let's get started. So in terms of reproduction, life has a few different ways in which that can happen. Some organisms reproduce asexually without sex. Asexually without sex. It's essentially sort of like mitosis, right? Where one cell produces another cell that's an exact copy or a clone, right? And we think of bacteria, prokaryotes. They do this a lot, what's called binary fission. And here's a little GIF or GIF, however you want to say, of that process happening, right? One cell, one bacteria divides and becomes two. Binary fission in bacteria. It's essentially, each of these is an exact clone of the other. Other organisms, like some single-celled eukaryotes, like yeast, for example, they go through a process called budding, and I'm going to go into that in a bit more detail in the next slide. Paramecium, amoeba also do this sort of budding-like reproduction. Now, simple multicellular eukaryotes, like a hydra, also does some budding. As you can see here, you can see the new baby hydra budding off from the parent. Now, the thing about asexual reproduction is that we're basically making exact clones, right? So there's little genetic variation, if at all, right? So this is easy. It's easy to reproduce asexually, but it doesn't give us as much genetic variation. And so here is how this process, yeast budding, occurs. You have a parent cell here. Here's the yeast, right? This is a eukaryotic cell. Remember, yeast are actually in the kingdom fungi, a single-celled eukaryotic cell. So here's our parent cell. And then the first thing that has to happen, you can see, is this nucleus here has to divide. So the nucleus divides. Half of it looks like it's going in here. We can see a little cleavage furrow starting to form. And then you see the two daughter nuclei in each of the cells. Finally, that last cell will pinch off, sort of like it was a little bud that came off of the original parent cell. So this is how many single-celled eukaryotic organisms reproduce. And the multicellular hydra, like we mentioned before. Well, what about the rest of us? What about a complex multicellular organism like humans? How do we produce? Well, I sort of hinted in the first slide. It's all about that egg and the sperm. Well, okay, making egg and sperm together, right? We get one from mom, one from dad to make baby. How does that happen? Can we do that by mitosis, right? Can we take one egg and have it undergo mitosis and make another egg? Well, let's think about that. So if your egg in humans has 46 chromosomes, right? In humans, we know we have 23 pairs, so 46 total. So if your sperm had 46, well, when those combine to make a baby, oh, that would be 92. We can't have 92 chromosomes in our cells, right? That would be double what we need. So something else must occur. We can't do mitosis for multicellular complex organism reproduction. So what has to happen is we need to reduce, for humans, those 46 chromosomes to 23 chromosomes. Now, other organisms will have different number of chromosomes, but the process is very similar. So we reduce the 46 down to 23. Basically, we need to go to half. That process is called meiosis. So if we take 46 down to 23 into the egg, 46 down into 23 for the sperm. When the sperm and the egg fertilize or come together, then we're back to our original 46. So meiosis is the process in the body that we make the gametes, the sperm and the egg. Most every other cell division process in our body, right? When you think of making hair or skin or all that, that's all, those cells are produced by mitosis. But egg and sperm are meiosis. There's a very distinct difference between these two. Okay, so I mentioned already that humans have 46 total, or 23 pair, of chromosomes. If we were to take those chromosomes and lay them all out during their metaphase state, right? So if we looked at them in metaphase and laid them all out, sorted them by length, you'd have 22 what we call autosomal, okay? So these are the 22 chromosomes that are not the sex chromosomes. And you can see sort of one's the biggest, we go down to 22, and 21, these are the smallest. This is called a karyotype. When we take and display the chromosomes from metaphase, sort them by length. So 1 through 22 are autosomal, and then the 23rd pair, right, the last two, are the sex chromosomes. Now, the most common number of sex chromosomes would be 1X and 1Y. We generally think of that as a male. Or 1X and 1X, we generally think of that as a female. But we know these can change, right? You can have an extra X there. Sometimes you have you can be missing a chromosome, right? So the sex chromosome numbers do change, and we'll talk about that in an upcoming video. But for the most part, what is most common is in humans these 46 or 23 pair of chromosomes. 1 through 22 are autosomal, and the 23rd pair is called the sex chromosomes, which determine sex or biological sex. So how does this happen? Well, if you think about an adult human, and we're going to focus on humans, we could be talking about other living things, but we're going to focus on humans. I think it's a little easier to understand. In our adult form, we are diploid, okay? So I mentioned that we have 2 of each, right? 2 of each chromosome. So we could say 2 times our number of chromosomes. 23 pair, so for our case, N is 23. And because right now as an adult, I have 2 of each of my chromosomes, I've got basically 46. So I'm 2N right now, or diploid, 2 sets. Now, what happens then, of course, is in the male testes, in the female ovaries, sperm, eggs are produced. That process of producing the sperm and the egg is called meiosis. So we take our diploid number, 2 times our number, and cut that in half to N. So for humans, that's 46 down to 23. For different animals, you would just divide by 2 to see what you get. So meiosis is the process of reducing that chromosome number into the sperm and the egg. Now notice the sperm is just 1 N, and the egg is 1 N. We call that haploid. Okay? So it's not diploid now, it's haploids. One set of chromosomes, 1 set of chromosomes for the sperm and the egg. Now, of course, fertilization occurs. The sperm, one actually gets in, breaks in, and that essentially is called fertilization, and we go back to our double number of chromosomes. And then, of course, the zygote forms, that's 2N or diploid fetus, and then eventually, of course, we get back to the diploid human. So this is how it alternates sort of through the generations. Meiosis cuts it in half. Fertilization restores the number back to diploid. And the process right there finishes out. Okay, so I'm going to use a vocabulary term here, homologous chromosome. If you think homo, you think same. So homologous, same information chromosomes. So I mentioned that we had 2 pair, right? So if, let's say, these 2 chromosomes here are your number 1 chromosome. You got 1 of each, right? These are your number 1 chromosomes, 1 here and 1 here. These essentially have the same genes. They carry the same genes, right? Maybe one of the eye color genes is there. Maybe one of the genes to make a certain enzyme is there. So on number 1, these chromosomes carry the same genes. Now, they might have different versions of the genes, which we'll get to later, right? One for blue eyes, one for brown eyes. But we call a homologous pair the same chromosome that has the same genes, same information. So if you think about it right here, okay, so we've got this organism here, it just happens to have 2 chromosomes. So 4 total, 2 pair, I should have said, 2 pair of chromosomes, 4 total. All right, so it looks like this one and this one go together, this one and this one go together. Okay, so these two would be a homologous pair. Same information. They're both number 1 chromosomes. Now, when those get ready for meiosis, what we'll see is those actually double. So we still have a pair. Here's one, and here's another number 1 chromosome. But they're going to duplicate, and we call this a double-stranded homologous pair of chromosomes. And you'll see that play out in meiosis. So meiosis is a double division. When we talked about mitosis, there was just one division. But in meiosis, we see it's actually a double division. So the first thing that happens is DNA replication, like I just mentioned. So we start in this cell, we're looking at 2 homologous pair of chromosomes. So let's say the blue ones are number 1, and then this is number 2. So we've got some organism that has 2 linear chromosomes. Okay, so the first thing, those have to duplicate. And so what you'll notice is they have duplicated now. So we still have the number 1 chromosomes there. We still have the number 2 chromosomes there. This is what prophase looks like. We see the chromosomes. Now, meiosis 1 happens, and what you'll notice is that meiosis 1 separates the homologous pair. So this was a pair, that was a pair. Notice one of them goes over there, one of them goes over there. So we've separated the homologous pair. Then those cells divide. All right, go through all of meiosis, and I'll go over the details of that in a minute. But in meiosis 2, notice that the sister chromatid, so there's a chromatid, there's a chromatid, those separate in meiosis 2. So the second division of meiosis is separating the sister chromatids. And what we end up with is 4 gametes, right, egg or sperm, that are each 1 N. Notice what we started with was 2 N, right? There's 2 of each, 2 of those, 2 of those. We ended up with 1 of each in the egg and the sperm. Okay, so important thing to remember is that meiosis is 2 divisions. The first division separates the homologous pair. The second division separates the sister chromatids. So let's look a little more deeply into meiosis 1. First division separates the homologous pair. This is what I just mentioned there. So what happens is they line up, they start to, you see the chromosomes in prophase 1. We're still 2N, right? And then what happens is in prophase 1, you'll see that this homologous pair that are already double-stranded will kind of overlap. We call this event synapsis, right? That's when the chromosomes start to overlap a bit. And it forms what we call a tetrad, right? Because we see each of the homologous pair, there's one of them, there's the other one of the pair, each with their double strands, right? That is called a tetrad because really there's essentially 4 chromatids. 1, 2, 3, 4. So we call that a tetrad at that point. Metaphase 1, they move towards center. And then telophase, you can see that the homologous pair separates. So remember, meiosis 1 separating the homologous pair. Notice this is the event where we go from 2N to 1N. We have separated the pair, right? So now this is 1N and this is 1N, where these, we had 2 of each chromosome. Meiosis 2 then will separate the sister chromatids. So as you can see, we're down to 1N already, but now we need to separate these chromatids. Okay, so these things need to be separated. So in metaphase, they line up in the center like normal. Telophase, they break apart. Now you'll notice I've got 1 of each, all right? So let's say that's our number 1 chromosome, that's our number 2 chromosome. We've got 1 of each, same thing. There's one of the number 1 chromosomes, whoops. Number 2 chromosomes, you can sort of see these are single-stranded, but 1N or haploid. Okay, so now let's go over the details of how this works. So meiosis 1 and then meiosis 2. So meiosis 1, as you'll remember, starts with prophase 1, then metaphase 1. So let's look here. Notice that we've got some homologous chromosomes. So in this case, we've got an organism that looks like 3 pair, right? So let's say that's the number 1 pair, the number 2 pair, and the number 3 pair. So as you notice here in prophase 1, we see the chromosomes. You'll also see that the centrosomes are starting to move to their opposite end of the poles. The centrosomes with their little centrioles are creating these little spindle fibers that the chromosomes will start to move down, as you see here in metaphase. Now, they attach, these chromosomes attach here at the centromere, and will start to move towards the middle. So in metaphase 1, we see they're kind of lined up right there in the center. Then, in anaphase, they split apart. So notice that the sister chromatids are still intact because remember meiosis 1 is separating the homologous pair. Then, telophase 1 cytokinesis, we still have two different cells that we're starting to see here, telophase 1 and cytokinesis. We're starting to see the cleavage furrow start to form in there. But now notice, we only have one of each, right? So there's one of the number 1 chromosomes, one of the number 2, one of the number 3, or maybe I said that backwards. But you can sort of see that we've got one of each. So in this case, we're haploid at this point. Here we started 2N, or diploid. Okay, so we're left with, at the end of meiosis 1, 2 cells that are both haploid. Then meiosis 2 happens. So we go through the same thing again, but this time we're separating the chromatids. So because we're left with 2 cells at the end of meiosis 1, notice everything here is times 2. So we see prophase 2, there they are hanging out. Metaphase 2, they've lined up in the center. Anaphase 2, notice that the chromatids here split, and now they're back into original chromosomes that we have. Each of these 4 cells, 1, 2, 3, 4, is essentially haploid, or half of the original information. We would say 1N. Now notice we had cleavage furrow here as well because these are animal cells, and we're left with either 4 egg or 4 sperm cells at the end of meiosis 1 and meiosis 2. Now I want to go into a little more detail of prophase 1 because a very important event happens during prophase 1, something which we call crossing over. During prophase 1, the homologous pairs actually swap some of their information because the chromatids intertwine. So if you look here, we're in prophase 1, right? You see the nuclear envelope starting to disintegrate. We've got the chromosomes. And then we start to see some synapsis, or the crossing over of those chromosomes. And notice right here, we see the full crossover begin. You see how some of this pair crosses over with this? Is that a big deal? Is some of the information from one chromosome getting on another? Yes. Actually, they swap, but it's okay because remember, these are still, let's say, number 1 chromosome, right? So it's the same, they're a homologous pair, it's the same information, it's just going to increase a little genetic variation because this one originally had this information, which is now going over here. So this will increase some of our genetic variation. The point of crossing over is called the chiasma. I've also seen it as the chiasmata, but that's where the crossing over occurs. This increases genetic variation by giving a new combination of traits. These sort of pinkish chromosomes now have the allele, or that version of the gene that was originally on that chromosome. Okay. Then we also want to talk about random fertilization. If you remember at the very beginning, I started with the picture of the sperm and the egg. So if you take any 2 parents, each with 23 chromosomes, right? They're each donating one of their pair, and multiply that, you essentially can have a baby with 70 trillion different diploid combinations. And I've got a picture here, a random family I found on the internet. And you can see, even within the same 2 parents, these 2 people, notice the kids. There's some genetic variation. It's got some blonde hair, this looks like it's got a little auburn or red hair. So depending on that random fertilization, what random sperm got with the egg, then you could have many different combinations. And as we mentioned, all of these random sperm fighting to get into the egg, one does. That's you. And so I always like to think of it as, what would it have been like? What would I be like if this sperm had got in instead of that sperm? Maybe completely different. Okay, so let's wrap things up here with a discussion of genetic variability. We started the whole video with the idea that mitosis occurs in bacteria and in yeast, and we basically get exact clones of each other. Well, that's not what happens in multicellular eukaryotic organisms like us. We have variation. So this process of meiosis gives us a lot of genetic variability, which we know is good for the species. So there are 3 ways I want you to remember from this video of how we get genetic variability. The first is that the homologous chromosomes in meiosis 1 assort themselves independently of each other. So here's your homologous pair, right? Here's another homologous pair. These assort independently. So you might get this one coming over here and this one coming over here this time, but the next time you get meiosis, those may have arranged themselves differently, and this one would actually go into the different chromosome with that, or into the different gamete with that one. So these chromosomes assort themselves independently. Second source of variation that we mentioned was crossing over. You see here, again, in prophase 1 that this crossing over, so this chromosome gave a little bit to that chromosome gave a little bit to that, and they swapped. And that increases genetic variation. And the final one that we mentioned, of course, was random fertilization. The random egg getting into the random sperm getting into the random egg increases genetic variation. So as you think about all of this, all of meiosis, what you need to remember is that meiosis is the process of taking us from a diploid organism, right, 2 times the number of chromosomes, to haploid organisms in egg and sperm.